First, I would like to thank the organizers for allowing to present our work on phenotypic properties influencing HIV transmission fitness. Sexual HIV transmission requires the penetration of the virus across the mucosal barrier and the establishment of infection target cells. And in the vast majority of cases, out of the pool of viruses from the donor, only one infectious clone or a small number of viruses establish infection. So to better understand the infectious process, we need an in-depth understanding of the phenotype of the virus that gets transmitted and how this may contribute to the genetic bottleneck selection process. So our study aim was to explore transmission fitness and analyze possible phenotypic differences between acute and chronic HIV-1 that may affect the genetic bottleneck selection process. So we collected a number of acute subtype B samples from the CHAVI consortium and also chronic subtype B samples from the Institute of Tropical Medicine in Belgium. And then we created envelope chimera viruses by yeast-based cloning of the envelope gene into an NL43 backbone. And then we performed a series of assays to look at these acute versus chronic viruses. Here we can see the acute versus chronic envelope chimera for their replication kinetics across potential target cells, including PBMCs, macrophages, dendritic cells, or T cells. And we cannot see really a difference of acute or chronics for any preference for one specific cell type, but chronic viruses seem to replicate a bit better in PBMCs and in T cells. So we also looked at replication fitness in PBMC cells, tested the sensitivity to entry inhibitors, entry kinetics, as well as entry efficiency. I'm not going to show you all the data, but I can summarize that we have not seen any selective advantage of acute over chronic viruses or vice versa. So to explore now transmission fitness, we have established an virus mixed infection model using human cervical and penile explant tissue. And we mix 4 to 5 acute viruses with 3 chronic viruses, and each acute virus is competed in every possible combination. And then we add the mix to cervical or penile explant tissue blocks for 2 hours, wash it off, and then after 2 days, we also collect the cells that migrate out of the tissue and co-culture them with T cells to look for infection in trans. After a 10-day culture, then we lyse both the tissue as well as the migratory cell T cell co-cultures and harvest the DNA for analysis. We perform then 5-4 deep sequencing and use it as a kind of counting tool that allows us to monitor each of these genetic distinct viruses within the mixes we have set up. So here's the result, what we see after 10 days in culture in cervical tissue. And we have in the top panel the tissue itself. And what we found was that the chronic viruses here seem to be stuck in the tissue where they replicate, while the acute viruses are mainly found in the migratory cell T cell co-cultures, so they seem to be the ones being pulled through and transferred to T cells. We see very similar result using penile tissue. Again, the chronic viruses seem to be trapped in the mucosa where they replicate, while the acute viruses are pulled through and transferred to T cells for infection. This slide shows a summary of transmission fitness mixed virus infections, because they're all done in replicates, and again, it shows in the top panel the tissue itself, and we see the chronic viruses, the blue bars, over-replicate the acute viruses, while in the migratory cell T cell co-cultures, the acute viruses are mainly found. And to do a transmission fitness experiment, we set up more than 100 competitions. And this slide just shows an accumulative analysis of these. And again, it quite clearly shows the chronic viruses are trapped in the tissue, the black bars where they replicate, while the acute viruses are taken up by the dendritic cells and transferred to the T cells, the gray bars. So we were now wondering or thinking, is there a relationship between the envelope glycosylation, lectin found in the tissue, and the infection process? So we know the model of DC sign capturing virus for antigen presentation and potential transinfection, and that's what we believe happened in our migratory cell T-cell cultures. But we also found in the literature evidence that lectins might have a different role in the infection process. For example, for other infection models, extracellular mannose binding proteins in tissue and C-type lectins expressed on epithelial cells have been shown to trap pathogens with high mannose oligosaccharides. Also, there was a very nice study which has shown that a homozygote carrier for a variant of mannose binding lectins, allele that decreased the mannose binding lectins concentration, that these people were at higher risk of HIV infection. So we now looked at replication fitness of the viruses in the tissue and compared it to their lectin binding affinity. And what we saw is that the viruses which were able to stay in the tissue and replicate, that this kind of correlated with also higher C-type lectin binding affinity. And then we also looked at the conserved end-link glycosylations on the envelope, and we found that all of our acute viruses had reduced numbers of end-link glycosylations compared to the consensus sequences of different subtypes. So, and at last here I want to show that we also have some in vivo evidence of mucosal trapping in the mucosa. And here we have a cohort of Ugandan and Zimbabwean women which were recruited within 3 months of infection. And we were able to get endocervical swab samples as well as plasma samples. And genetic analysis showed that in the endocervical samples, we have a high genetic diversity of virus, while in the matched plasma is a homogeneous population. So to summarize, HIV-1 genetic diversity may be higher at the site of infection than in the plasma at early infection, at least in our cohort. And our ex vivo transmission model shows a barrier which is highly dependent on trapping virus, high glycosylated virus, while virus with fewer glycosylation is preferentially taken up and transferred to T cells. So in conclusion, HIV-1 with reduced glycan shield may be passively selected for the transmission by escaping a lectin trap. So I would like to thank my mentor, Eric Arts, and all the people who were involved in this study. Special thank also to Robin Shattuck at Imperial College, with whom we have a very strong collaboration with the tissue model, and also thanks to all of the funders. Thank you very much.